thank everyone for joining my talk today too, and the organizers for bringing together such a comprehensive discussion for all of these emerging spatial genomics technologies. So today, as he was mentioning, I'll be talking about the spatial genomics platform we've been working on here at Vizgen. I'll first introduce the technology, and then I'll highlight some of the advantages of a high-quality spatial genomics measurement by walking through a couple data sets. But this overall, I think it's really going to be an exciting year as we see these sorts of spatial technologies become mainstream research tools. But as a scientist, I'm really most excited to see what researchers can start to do once they have these tools in their hands. What new biological discoveries it will enable, the impact it could have on the impact it could have on understanding human disease and their treatment. But before we get too carried away speculating about the future, let's look back at the beginning of genomics, maybe 20 years ago. When we look back, it's really amazing what's been achieved in just 2 decades, and it highlights the power of being able to measure biology with tools for highly multiplexed measurements that begin to match the complexity of these biological systems. There's tens of thousands of genes in the genome, and to understand how the tissue is working, it's useful to measure as many of them as possible. From bulk sequencing, we've been able to understand which genes are expressed within each tissue and how that global gene expression changes with disease or treatment. More recently, with the widespread adoption of single-cell dissociation techniques, we've been able to understand gene expression within individual cells. This lets us cluster groups of cells based on their gene expression patterns and define these stereotype cell types and cell states. However, biological systems are also intricately spatially organized. When the tissue is ground up or the cells are dissociated, we lose all of this spatial information. The next generation genomics is going to add the spatial component back. And instead of dissociating a tissue, grinding it up, or breaking it into cells, we're going to be directly identifying hundreds or thousands of biomolecules within intact tissue slices. So then we can begin to map out the functionally relevant organization of these biological systems. And that's what MERFISH is. MERFISH, or multiplexed error robust fluorescence in situ hybridization, is a technology that builds upon single molecule FISH. So in single molecule FISH, fluorescent probes are designed to bind specifically to a target transcript, and then the sample is put onto a high resolution fluorescence microscope, and the fluorescence from each molecule is imaged onto a camera. Each individual RNA transcript then shows up as a diffraction limited spot in the image, and the gene expression can be quantified by counting the number of these spots that appear. But due to the spectral overlap of fluorescent dyes, single molecule FISH is traditionally limited by the number of distinct colors that can be imaged at once. MERFISH greatly expands this multiplexing capacity by encoding each gene with a combinatorial barcode and then reading out that barcode using sequential rounds of single molecule FISH. By decoding the pattern of brightness and darkness at each position across the sample, we can identify exactly where each of the targeted transcripts is and achieve this very highly multiplexed transcriptome-wide spatial measurement. So MERFISH as a technology has already been around a few years. It's originally published in 2015 in Xiaowei Zhuang's lab, and over the past 6 years, there's been a number of biological demonstrations, both coming from her lab and even from other labs with the growing adoption among the broader community. Some of this is organic, where the researchers have read the scientific paper, collaborated with one of the researchers developing MERFISH, and built up the ability to perform the full measurement on their own. But a growing fraction of these users are already enabled by Vizgen, and we're starting to generate their own publications using Vizgen instrumentation. So this includes even 10 institutions that I didn't even list on this slide that already have MERFISH data in their hand through Vizgen's lab service and early access programs. So within Vizgen, we're really making MERFISH more broadly available through our commercial solution called MERSCOPE. Just last week, we announced the commercial launch of our MERSCOPE platform, and this platform facilitates the full MERFISH measurement with a complete reagent, instrumentation, and software solution. So MERFISH is able to combine the advantages of MERFISH technology with a streamlined workflow. Using MERFISH technology as a foundation, we are able to maintain the very high sensitivity, the high multiplexing capacity that's already been well established, and we combine this with easy to use automated instrument and software tools to facilitate the design of custom gene panels for each measurement and data analysis and visualization. But it's really been an exciting time at Vizgen as we put together this platform. But ever since spinning out Vizgen from Xiaowei's lab at Harvard, there's really been one driving principle that has remained really most important for us. Since MERFISH already started with this reputation as the highest quality spatial genomics approach from all of the publications coming from Xiaowei's lab, we didn't want to compromise any of that data quality as we built MERSCOPE and made it more broadly available. With the engineering of the instrument, we've made really no compromises on data quality. We maintained a high resolution, high NA objective to be able to capture as many photons as possible. But as we developed the instrument, this has its own challenges. The lab-built microscopes used for all of the original MERFISH publications were built on these very large, bulky optical tables that aren't as conducive for a commercial platform. But as we engineered the instrument, we put all of that vibration isolation into the instrument. So we're able to achieve the same performance on it with an instrument that can sit on the lab bench. And it's really great as we have this instrument in-house. It's great seeing the quality of the data that we're getting off the MERSCOPE instrument, and I'm excited to get it into the hands of researchers. So as with any genomics research tool, MERSCOPE isn't limited to any specific biological application. It's applicable to nearly any biological question. This could be a neurodegenerative disease, where you can look at how cell state is changing around morphological indicators of neurodegeneration. It's applicable to cancer immunotherapy, where you can see how the interplay between drug treatment and tumor microenvironment is able to modulate the activation state of different immune cell populations. And you can look at infectious disease and understand the immune response in the vicinity of infected cells. Let's take a look at the data. I think a natural place to start is with the standard benchmarks for spatial technologies these days, which is the brain. Since so many technologies are first demonstrated on the brain, it's a good system for understanding the strengths of MERFISH technology. So here we put together a gene panel of 483 genes. This includes about 50 canonical markers and around 450 cell receptors, including a lot of GPCRs and RTKs. And we use this gene panel to measure a full kernel slice using the MERSCOPE instrumentation. Here we're just looking at 8 of the 483 genes, since it's impossible to plot all 483 genes simultaneously. But you can already see the rich spatial heterogeneity across the sample. But MERSCOPE measures the full tissue slice with single molecule resolution. Each transcript is localized with better than 100 nanometer precision, and we can zoom into any portion of the tissue to see exactly where each transcript is localized within the intact tissue. Here we're showing every detected transcript from all 483 genes and drawing the segmentation boundaries around each cell body. For any high-quality spatial genomics measurement, it should be quantitatively consistent with established methods, and it should be highly reproducible. MERSCOPE is able to meet both criteria. When we compare the copy number of each gene detected with MERFISH to the FPKM from bulk sequencing, we find strong correlation with no leveling off or change in slope, even as we look at the most lowly expressed genes. When we compare the copy number between replicate MERSCOPE measurements, we again achieve very high correlation and consistency in the number of counts detected. But what does high quality data mean for using MERSCOPE to answer biological questions? By building upon single molecule FISH, which in many cases is the gold standard for RNA quantification, MERFISH maintains the highest sensitivity and resolution. From single cell transcriptomics, we know RNA expression is already relatively sparse. Most cells are expressing a few tens of copies of each transcript. And since RNA copy numbers already start out relatively low, it's really critical to be able to count as many, if not all, copies of each transcript within each cell. Just to highlight how this manifests in real data, we can look at a quick comparison between a MERFISH measurement and a measurement from an oligo array capture-based technology. So MERFISH is identifying the transcripts in situ, while the oligo array technology requires the transcripts to be able to diffuse onto the oligo array and be captured by the barcoded oligos sticking out from the surface, and then sequenced to determine how many of each different transcript was captured at each grid spot in the oligo array. So here we're considering match samples between the two technologies. When we look at the number of transcripts detected with MERFISH, when we look at the number of transcripts detected, we find that MERFISH consistently detects 70-fold more copies of each transcript. But what does this mean for understanding the biology? If we look at a moderately expressed gene, this opioid receptor, with MERFISH, we can see clear spatial organization. However, if we look at the same gene with the oligo array technology, there are only a few grid spots with 1 or 2 counts, and there's no clear spatial pattern. You can see that you really lose the biology that could be driving the importance of the expression of these different moderately or lowly expressed genes. And even if we look at the resolution with these grid spots, you have little to no information about individual cells, since each of the grid spots takes up the size of maybe 10 different cells. So although these oligo array based technologies can be genome wide and you don't have to design a predefined gene panel, they are in some sense biased. They completely miss these moderately or lowly expressed genes. Finally, for this data set, the single cell resolution of MERFISH allows us to perform single cell analysis and atlas the cellular composition of tissues. So by counting the number of transcripts within each segmented cell and clustering the cells based on gene expression, we can perform the same sort of analysis that you can with single cell sequencing. We can identify cell types, but this is a spatial measurement, so we can map those cell types back to the exact position within the tissue and create an actual atlas of the cellular composition of diverse tissues. So from this UMAP, you can also, if you look more closely, you can also see how we're just closely clustering the cells here, but there's a lot of rich heterogeneity within these clusters that we see. And there's many sub cell types that can likely be identified. So you can look more into some of the published MERFISH work just to understand how this high detection efficiency of MERFISH even allows you to detect neuronal subtypes that are even difficult to see with single cell RNA sequencing. So you can download this data and analyze it for yourself through Vizgen's data release program. If you're interested in looking at the data, you can sign up for it on our website, and then we'll send out an email once the data is available. So our first data set is going to be just like the data I'm describing here. It's going to be 9 full kernel slices using this 483 gene library, and it'll be live within a couple weeks. So we've measured these full kernel slices at 3 positions in the mouse brain with 3 biological replicates for each position. And we hope this can be a useful resource for the community. So for each of these data sets that we're providing, we'll include the standard output that comes from every MERSCOPE instrument run. So MERSCOPE automatically decodes the raw images coming off the microscope to get the results that are most biologically relevant. This includes the core spatial genomics output, which is the spatial position of all of the detected transcripts in the sample. This is tens or hundreds of millions of transcripts in each measurement. So along with this, we output high-resolution mosaic images of the additional stains across the whole tissue, such as DAPI to mark the nucleus and poly-T to mark the cytoplasm. But MERSCOPE image processing also performs cell segmentation so that users can immediately perform single cell level analysis. And the associated outputs of this cell segmentation include the cell by gene matrix, indicating how many transcripts from each gene were detected within each of the segmented cells. This is just like the cell by gene matrix you see from single cell sequencing. But since it's spatial technology now, we know the XYZ coordinates and volume of each of these cells, and this output is put into a separate CSV file for additional analysis. For even deeper analysis, we also output the full cell boundary polygons for each of these cells in 3 dimensions as separate binary files. But MERSCOPE doesn't just work on the brain. MERSCOPE also works on clinically relevant human tissues. Here, we're mapping the expression of 417 genes across a colon cancer section. And here we highlight 10 genes that mark different cancer cells, immune cells, and fibroblasts. With just 10 genes, we can see very clear cellular diversity and interesting spatial features across the tissue, just as with the brain. We can also zoom into any particular portion of the tissue because we have the full resolution across the whole tissue slice. And we can look at, for example, this group of SOX9 expressing tumor cells. So this group of SOX9 expressing tumor cells encircles this SPP1 expressing cell, and immediately adjacent, just below in this image, you see a large population of B cells marked by immunoglobulin alpha-1 expression. And just as in the brain, we can then quantify the transcription at a single cell level and use all 417 genes in this measurement to map out the cellular distribution. The spatial distribution of immune cell types and states can indicate how effectively the host is fighting the tumor. And here we see a rich variation in the distribution of immune cell types, with large clusters of B cells and other immune cells that seem to be infiltrating from the intestinal lumen, and T helper 17 cells highly enriched on the other side of the tumor. With MERFISH, we have a direct view into the structural composition of these cancer tissues. So as I mentioned earlier, we announced MERSCOPE last week. We'll have a pilot batch going out this summer, and we'll have full availability later in the year. We're building up the wait list for the summer launch, and if you're interested in applying MERSCOPE to your research, feel free to get in touch with Vizgen with the information on this slide.